Okay, thanks everyone for joining this Introducing KBase webinar. My name is Zach Crockett. I am a technical professional at the Oak Ridge National Laboratory in Tennessee, and I will be introducing KBase to you all and showing what you can do in KBase. So just to start off, if you are unfamiliar with KBase, KBase is a community-driven research platform for systems biology that seeks to empower researchers by integrating data and tools to help accelerate analysis, support collaboration, and help publish discoveries. So the KBase is part of the U.S. Department of Energy's Biological and Environmental Research Program. So we fit into the BER ecosystem to produce, analyze, and publish data along with other BER programs such as the Joint Genome Institute or the uh, Molecular Sciences Laboratory. And so the goal of KBase is to bring in this data from other research facilities in DOE or outside of DOE, and to allow users to analyze that and export the data for publication or to use in other platforms. And so this is a graphic of how KBase fits into that data lifecycle. So we provide tools to integrate data and create analyses, and share your analysis and data with collaborators, and then publish the results. And when you're working in KBase, the sort of the fundamental unit of analysis in KBase is what's called a narrative. And if these narratives are built on Jupyter notebooks, so if you're familiar with Jupyter notebooks, you'll recognize some of the elements. Um, the the core functionality of the narrative is here in this big section in the bottom right, where you can add cells. You can add a markdown cell to document what you've done. You can add a code cell to run some limited Python code, and you can access app cells to run apps that we have developed and have been developed by our collaborators to do analysis on data. And then finally, you can add a viewer for these data objects to get information about the different types of data you've produced and has been uploaded. And so in this orange highlighted section on the left is where you'll see all the data that are in your narrative. So this is
हाँ.
so these data are either uploaded by you or produced by the apps you create, or they can be copied from other users or public sources. And then in the bottom left is the apps panel where you will find all the apps that are available for use in KBase. These are Many of these are third-party apps that you may be familiar with, such as Quast or Spades, and some of them are in-house tools that KBase has developed ourselves. And these apps are designed to work together. And so for the apps, for example, for genome annotation, the output of any genome annotation app should be it is a data type that can be plugged straight into these taxonomic assessment and metabolic modeling tools. And so these help the different data types on KBase interact and facilitate running different steps of the analysis together without having to worry about file format conversions or moving data between different file systems. And this is a high-level summary of a more or less complete workflow of what would be found in KBase, but it's not comprehensive, and you would not necessarily need or want to do all of these steps in any single analysis. So in those apps, we have about 200, probably a little over 200, and we have them categorized by function, which I will show when we get to the live demo. And some of these some apps cover multiple functions. But this is just a pretty broad summary of what is available. If you have, if you want to see a full list of everything, you can view our app catalog. And if you are a tool developer, you may be interested in learning about the software development kit to add a tool you've created into KBase to be used by other people. So to get your data into KBase, most people start with their own data, usually some kind of basic biological data like FASTQ reads. And so you can upload those into the staging area, either through drag and drop straight through the browser, or use the Globus service for transferring large files. And our importer will try to guess the file based on the file extension, and that'll help you pick the right import type. Um, some files could be multiple import types. So as you see in this example, the FASTQ reads could be interleaved or non-interleaved. So you can choose the correct import type for your data. And you can import multiple data types simultaneously. So you could import both FASTQ reads and a GFF genome all in one go. We also have some data pre-populated for use from many of the major public sources, such as JGI or RefSeq. You can find these in the narrative and copy them straight in. You can find them in the narrative interface and copy them straight into your narrative for use in your analysis without having to download and re-upload these. And related to that, we, as part of the BER program, we are working with other BER programs to create a data transfer service that will allow you to directly import data from JGI, NMDC, ESSDive, and EMSL directly into a KBase narrative without having to download and re-upload. And this is something that's been in progress and is nearing a point where it will be released in the near future for users to import into KBase. And you'll need ORCID authentication to access this, which we will talk about later in this presentation. So when you're working in a KBase narrative, by default, all of your data and analysis are public, or sorry, are private. You can make them public by going to the share menu and clicking make public, or you can share it with select collaborators by username. And this lets you control exactly who has access to see your data or to add to it. Organizations are another way to manage sharing. So you can create an organization for your research group or your course if you're a professor, and you can add all of the members who need access to that data, and you can add multiple narratives into that. And so what this will do is give everyone that is a member of this group read-only access. So you can add the narrative once to your organization, and it gives access to multiple users rather than having to pick them all out individually through this previously shown screen. You can also create a public organization which gives everyone that access to see your narrative. So if you have a large collection of published data that you want to make available, you can create an org to organize it and then make that org public to let other users have access to see it and to view it and copy it into their own narratives. A new feature we've recently added within the past few months is collections. So these are curated collections of data from collaborators that make their large they're large sets of data, such as genomes or models available for all users. Currently, we have four public collections, which are Enigma, Grow, the GTDB genomes, and the plant micro interfaces published genomes. And so these will allow you to compare these large data sets with your data. So to compare genomes in your sample to, for instance, genomes previously found by Enigma or PMI or Grow, and to copy some of these other published data sets into your narratives. And this image is just a display of what the org look or the collection looks like. I will walk through it a little bit more later, but you can see an overview which tells you um, what is in the sample. You can see a map of where the samples were collected, and you can see tax accounts for the different organisms that are in this data set. And you can see there the genome attributes that were calculated as part of creating the collection. So these will give you, for this particular collection, it'll give you completeness and contamination for these metagenome assembled genomes or MAGs, and it'll give you the classification that was um, predicted for this MAG to let you filter through the high quality MAGs or all MAGs or MAGs by a certain taxa. And this is just showing the taxa count again. And for this collection, we also have available a micro trait table to see for the different mags. And then finally, you can see the sample attributes, which tells you where it was collected, the environment, who collected it, etc. And then once you are done with your analysis in KBase, hopefully you will want to publish it. And so what you can do at that point is create a static narrative, which is a publicly available copy of the narrative that is available outside of the KBase login. And you can use this in your publication as a data set. We can we can register a DOI for you through the Department of Energy's Office of Science and Technical Information, and that will give you a persistent identifier for your narrative to help it be found um, either inside or outside of KBase and to be available in a publication in your data availability statement. And as part of that, we are working with the American Society for Microbiology's microbiology resource announcements to streamline publishing narratives in their publications. And we have a couple of templates to help guide you in a genome announcement for isolates or MAGs if you have either of those data types to help you get through the KBase analysis and to be able to export your results already packaged up and almost ready for a MRA publication. And so here's just an example of one static narrative that has a DOI. So this group out of Brazil has did a large metagenome study in KBase, and they produced this KBase narrative linked here, and we got them a DOI for that narrative. So this DOI will direct you to this narrative, and then they cited that narrative in their publication. And this narrative has been reused a few times by different users to compare that published data set with their own. And another new feature that if you are on our mailing list, you will have seen in the last few weeks is we've added ORCID record linking, which will allow us to read and write to your ORCID records. So if you grant ORCID record link permission, when you create a DOI, we will in the future be able to push that directly to your ORCID record, like like many publishers do when you publish with them, they can push the work directly to your Orchid account. And so this is separate from using Orchid for creating an account because this will grant different permissions, and I'll show you how to create and revoke that record link later. Another feature that has been added in recent years is bringing in experimental data. So these are part of our samples service. So you can import a spreadsheet of metadata for your samples, which includes things such as geographic location, temperature, collection information like who collected it. I mean, things like that to provide context to your data. So for instance, import your, you could include your geographic location um, to compare mags from similar locations. And you can import your environment location to find mags from different environments. And you can link these all to data objects in KBase. So that way the sample metadata collection follows the data and to any subsequent processing that produces new data. And this is just an example of some of the types of data that are linked in KBase. So the the main the main connected data types to samples that provide value added apps are metabolomics and chemical abundance data, metagenomics, and OTUs from amplicon data. And so you can import these data types. You can import these data types or produce them from other sources, and then import your sample metadata, and then collect them, and then get derived products that are based on both the data and the metadata. And the goal of this long term is to connect data types across scales and across systems to compare different. You may have some or all of these types of data, but we want to be able to connect them across users and across data sets. And then this brings us to our KBase Educators program, which is led by Ellen Dow, who's on the call with us. So this was started about 5 years ago during the COVID pandemic and has really grown to, I think, over 200 educators now. And so if you're an educator, you can join the program. You can find teaching materials that have already been built. You can adapt them for your own class and let the community know how it worked for you. And you can update and create new workflows to share with the community and work together with other educators to share resources. And part of this that has been developed by this group is a collection of modularized workflows covering many of the sort of core functionalities in KBase, such as genomics and metagenomics. And these are all, most of these are linked together, so that way you can connect these different broad workflows to each other. And a collection of these has been translated by one of our interns, Malin Scott. So we have the ones that have stars are available in Spanish as well as English. And so this shows you a little bit about who is in this educators community. Over 270 members from 27 countries and 238 institutions. And as you can see, a lot of these are also um, undergraduate community college and high schools that may not have the resources of a large research university. And so KBase provides support for those users and educators to teach bioinformatics using this free resource. And finally, this is just a snapshot of where KBase stands with respect to um, user count. We have over 47,000 total accounts. We get about 2,000 new members a month. We have over 100,000 user narratives and over a petabyte of total user data. And you can find us on social media on LinkedIn, and you can find our YouTube at DOE KBase for recordings of webinars and other presentations. And this is just a picture of the KBase team from our retreat a couple years ago in Berkeley, California. We have um, we have people from four labs: Berkeley Lab, Oak Ridge National Lab, Argonne, and Brookhaven National Labs. And if you are curious about learning more about the origins of KBase and the details, you can find our project paper here. Okay, and so now I'm going to move into a demo of KBase features. But before I do that, are there any questions? Let's see. Okay, yeah, so I'll show about downloads in the demo. To answer this question, we do not have single cell RNA-seq at the moment. That's something that I think we would be interested in adding, but it's not in right now. Any other questions before I go on to the demo? Okay. So to start, what you'll want to do is go to kbase.us. This is our public-facing homepage. This where you can learn a lot about KBase. So we have our Learn page has a lot of summary about KBase, and it has some tutorial narratives that have been created and by us and by third parties. But when you're ready to jump in, what you'll want to do is either sign up or log in. And so if you haven't created an account, and you'll sign up, and you will get to this narrative navigator page. When you first create an account, all this in this main section will be blank, but I have a few narratives on this account. And so you can see the narrative section of this has four tabs. So my narratives are the narratives that are owned by you and created by you. Shared with me narratives are narratives that were created by other users that they've granted you access to. Tutorial narratives are a selection of curated tutorial narratives that demonstrate different workflows within the system. So we've got microbial genomics, we have some viral analysis, we have metabolic modeling, and RNA-seq. And then the the public narratives are all narratives that were created by any user that they've made public. So you can see Ellen has a few here, and you can search for other users if you know their username. So like my main account, I have several narratives. You can also search by narrative title to find the ones that are available. And when you're ready to create your own narrative, what you can do is click on this new narrative button over here in the top right. Okay, and so when that is ready, you will be able to see that there's a welcome cell, which has links to documentation, a link to the data and upload and download guide, a link to running apps, and then a link to some of our common workflows, as well as the terms and conditions, which you will have had to agree to when you created an account. If you notice this little eye icon here, that will let you toggle between view only and edit mode if you own it. So if you open a narrative that you don't own but you have access to, you have view access to, such as a public narrative, it'll look like this. You can copy the narrative to create your own copy that you can then interact with. Um, but if you own it, you just click it again to enable edit mode. It's always good practice to name your narrative when you first start it, so it will not exist just as untitled. And then once you have it named, you'll be able to manage sharing, and you can make it public or you can share it with another user. For instance, I'll share it with Ellen, who's on the call, and so now Ellen can view it, and I can also share it with another user. And so my other account, I will grant edit and save. So now my account, Z Crockett, has access to this narrative and can edit it and can add data and apps. Ellen can see it but can't add data or apps. But she can copy her own to then fork off of mine. And so the next thing you'll probably want to do is to add data to the narrative. And so you can click on this Add Data button. And so you'll see several tabs here. So my data is data that you've already um, added to another narrative. It will list the type of, or sorry, the name of your object, the type of your object, the narrative where it's located, and it will tell you when it was last updated and by whom. And if you want even more information, you can click on the white space to expand it, and you can click the binoculars to view it, the the data viewer in another tab. This will give you, the data view will give you a summary of the data. In this case, a sample set has a summary. It has this list of samples. And if you include the geographic coordinates, it will have a map. And then you can see the sample spreadsheet, and you can find other data sources that are linked to it. So this will vary by data type. Different data will have different tabs in the data view. So for instance, genomes will have assembly and annotation information, and it'll give you sources for the annotation and predicted functions. But the samples will usually just have broad overview of where the sample was collected and what is in it. You can see the object overview for a history of the object, so it may have different versions. In this case, I only have version 1 of this, but if I write over it, I'll get a version 2, and then I can view the archived version 1. And the provenance graph will tell you what objects it's derived from. With an upload, there won't be any. But if you have reads that you assemble into um, an assembly, then it will show you the reads that were assembled into that assembly, and then if you annotate it, it will show that that assembly was annotated into a genome. And then the related data and link samples are available for sample sets, and they just tell you the data that is connected to that sample set. So if you want to start working on something, um, you can find a data object to add. And so I'll just add this genome into my narrative. And so now you can see it's available in the data tab. If I wanted to find um, a genome that was shared with me by another user, I would go through it the same way. This account doesn't have any shared narrative, so there's no data to add here, but you can see um, The interface is the same. It just sorts them between the data that's owned by you and shared with you. The public data tab is where you can find the data from the sources I mentioned before. So you can find NCBI, microcosm, phytosome genomes. You can find public media. You can find some RNA-seq reads and some plant resources. So likewise for these, if you want to add one to your narrative, you just find the one you're looking for and click Add. And then the example data has just a small selection of most of the different data types. So these are generally pretty small data sets. So like these particular reads are a very small read set, but you can copy these just to find what apps you can run on that data type if you have that data type of your own. And then finally, probably the place you will be most likely to go is this import. And so this is where you can bring in new data that you own. And you can do that either through a drag and drop interface or through Globus. So in this case, I've just got these two small files that I'm going to import directly through the drag and drop. And so those are now available in my staging area. So this is a temporary file storage that will last for 90 days to where you can upload files that you can then import into your narrative. So for this case, I'm going to choose the type GFF plus FASTA genome. So these two go together. I have the assembled genome in the FASTA file, and I have the annotations in the GFF file. And so in this case, because I chose two types that go together, it's going to ask me which file goes in which place. So I'll select that the annotations go with the FASTA and the GFF goes with the GFF. And I'll give it a name and um, choose the parameters that are applicable. So I don't have a scientific name for it, but it is a draft isolate and it's from JGI. And so I don't have any of this other information, but it's not required. So I'm going to go ahead and click run. And so that will go through different stages. It'll first start off in queued, and this one went pretty quickly to running. So you'll be able to click on the line for this particular job in the bulk import cell, and then view the logs to see how it's progressing. And so this one is running pretty quick, and it's already uploading the assemblies. So this will probably run for a minute or two, and then it'll create new objects in the data panel. And so those objects are now created. So you'll see when it imported this, it created an assembly for the FNA file, and it created a genome, which is a combination of the FNA file and the GFF. So if you click on the names of these objects in the data panel, it will add a viewer to the narrative. And so you can see some information about the object. So in the case of a genome, you will see the size, the source, and then you can browse the features. So you can see the different annotations that are included in this. Um, you can see where they're located, their length, and if you want to see more about the particular feature, you can actually click on the name of it, and it will pop up another tab. And so it'll tell you the feature ID, alias type, the function, and it will give you uh, a protein translation and the DNA sequence. For assembly, it's a much simpler data type with less in it, and it simply tells you the contigs. So you can find a list of the contigs and their length and their GC content. So now that you've got your data uploaded into KBase, what you'll probably want to do is run some sort of app. And so I have a genome here. And so if you click on the white space or on this little three dots icon, it'll pop up at this expanded view of it, and it'll give you a quick summary of the object, but you'll see a lot of buttons here that have some pretty handy functionality. So the binoculars do, as I demonstrated earlier, they'll open up a data view where you can see more about it. And you can see the same type of information, but as you see, it's different for the genome. You can find the taxonomy information, which I didn't assign, but you can if you have it available. If you do have a taxonomy, then it will search publications to find related publications to that taxa. And then you can view more information about the annotation, such as finding the seed roles that were matched to these annotations. And so back in the narrative, the let's see, one of the handy functions is if you name over an object, if you create another object called test genome that is not the same data, what it'll do is it'll increment that and it'll give you a version 2 of that. And so if that was a mistake, what you can do is click on this little clock button to view the history and revert the changes if you made a mistake there. This little dendrogram icon takes you to the provenance view that I showed before, which you could also get to through the data view. The download button will let you download the object to your computer if you choose the options GenBank or GFF, or it'll let you add it to your staging area if you have a bunch of these files or very large files you don't want to download directly through the browser. And then the JSON representation is it varies by file type, but it's the structured JSON information about the object in the workspace. So this will provide some metadata about the object, and you can get a lot of the data out of this JSON, but it's not necessarily the same information as you get from a GenBank or a GFF. And then if you want to run an app on it, what you can do is click on this left-hand button to show apps with this as input, and that will automatically filter the app cell to let you find only apps that are built to accept that as an input. So one thing you can do with a genome is to build a metabolic model. So I'm going to use the newer model seed app, MS2, build prokaryotic models. And that'll open up a new app cell. And if you noticed, I had this import cell selected, and so that it put the app cell below that. And so you can either move it down, or you can just select the last cell in the narrative you want to add to the bottom. I'm going to delete this one because it's a duplicate. So to do that, you'll click the three dots and then find this delete cell button. And so this app will let me build a metabolic model from this genome. So to do that, I'll have to choose the mandatory parameter, which is indicated by the red line here. And so I'll add a new space to add a genome. And choose the one that I want to use from the dropdown. And you can see in the display here, it says genome type. So any type of dropdown parameter will only let you select the accepted types by that app. So in this case, it is you can find that information if you click on the spec. If you click under the genome and then click spec, it'll bring up what this app expects. And so um, the item here that we want for the genome or genome set, it will accept types, KBase genomes, genome, or KBase search genome set, which matches this data type here. So that will let me select that type. And then for this one, it gives a default suffix. And so this one is now ready to run. And so I'll click run and I'll let that run for a while. If I don't want to wait on it, I can close the narrative and come back later, or I can go back to the home page and click on the jobs tab. And this will give me a list of jobs that I've run over the selected time period, which can be hour, every 2 days, week, month, year, all time, or you can set a custom range. And so you can see I've had in the past week, I've run 2 jobs on this. So this one is 2 real jobs. This is a parent job for the import that I ran. So I imported the file as GFF, and then sorry about that. And then I ran the MS2 app, and you can see that that's running. It's been running for under a minute. So it may be done soon, or it may take a few minutes. And so when you're ready to complete your analysis, you can share the narrative with everyone. If you want to make it available as part of a publication, you can click Make Public. And so now this narrative, if you have an account, you can find this narrative the in the Public Narratives tab. But if you want to make it even more available, you can click Share and then Manage Static Narratives. And this will allow you to create a publicly available version outside of KBase. And so you can make that available outside of KBase for anyone, whether they have an account or not. And so they can see it based on, so this link, these kbase.us/n/ links are available without logging in. And you can see an overview of the narrative. Some apps will have viewers that can be viewed in outside of KBase, but if they don't, if the data relies on KBase services actively, it'll have a link to be able to access those viewers. And then you can also see a summary of the data objects that are included and what types they are. And you can see an automated citation list for all the apps that were used. So these are the 5 apps cited by the MS2 app. And so you can add these as citations to your publication. And so that was a very quick demo of the functionality and how you would go from a brand new narrative to publishing a narrative. So if you want to publish a narrative, one thing we recommend for the future is that you do this ORCID record link. So you go to account and then ORCID record link and click on this button to create ORCID record link. And it will take you to the ORCID authentication. And I can't do that on this account because it's already tied to my new account, but you'll just sign in with Orchid and grant the authentication. And in the future, we'll be able to directly add these DOIs to your Orchid record. And then I'll show the collections here. So these are in the main narrative navigator window in the bottom left. You can find collections, and you can click on any of these to see a summary of it. And if you want to interact with it, you can, for instance, go to the genomes and then match my data. And I don't know if I have data that matches this. So you choose a rank in a GTDB lineage to match, and then look for your data in one of your narratives and find the data object you want to match, and then create match, and it will try to match your genome with one of the genomes in the set. But I can't do that for that one because my object does not have a GTDB lineage assigned. And then I think the last thing I'll show is the catalog. So this is where you can find the full list of apps. So if you want to, they're organized by category. So some of the apps are across multiple categories, but these categories should cover pretty much all the types of analysis that can be done in KBase. So you can either scroll through and look for it, or you can search using the search bar at the top. And if you want more information about the app, you just click on its little card and it will open up the app's catalog page where it will have a summary of the app in general at the top. So this will tell about what the app is and how it works. Then you'll see the specifications for the app that'll tell you the inputs that it accepts, the outputs that it produces, and the parameters that you can set for this app. And then below that, it lists the related publications that this app cites. And finally, it's got a link to the GitHub repo for this app where you can see the publicly available code that is used to create this app. Okay, and so that is a general overview of the system. So I will move on to the Q&A doc, and it looks like most of the questions are answered. Okay, so the first one is about uploading a FASTQ file. So FASTQ reads interleaved is for FASTQ reads that have forward and reverse in the same file. FASTQ reads non-interleaved are if you have single end reads, such as from like a nanopore sequencer, or if you have paired end reads, but they're separated into a forward and reverse file, you can use the non-interleaved format for that. So interleaved just means that the actual forward and reverse reads are interleaved within the file. So the file alternates between forward read and reverse read. For this question about long read sequencing data, so we don't have I don't think our tutorial on that is available yet, but that is one that we are working on, and we do have some apps for that. Where's my narrative? So we have Fly for assembly will take long reads. We have Built Long for filtering reads. We have polypolish for polishing the genome assemblies with short reads if you have them. And we're working on adding a couple more. And I'll also point you to our YouTube channel where we have a webinar on this. We have this long read sequencing and assembly workshop webinar, which I will add to the Q&A doc. So that has a more comprehensive summary of the long reads tools that are available on KBase. Okay, and so this question, yeah, I should have covered this question at the start. So KBase handles the tasks by app. So how much resources the app is allocated is determined by us for each app. So if you're in the jobs, if you're in the job browser, you can see where it's running. So we have these different servers with different amounts of resources. So we have NJS, which is the main one, which is not, I don't off the top of my head know exactly how much CPU and memory it has, but it has a moderate amount. And we have a big memory queue. We have a extreme queue, which has even more memory and more CPU. So that is all determined by the job execution engine. And so it can be modified if you're running into limits. If your job isn't being given enough resources, we can modify the app to request more, and that happens fairly often, but not all the time. So I think for the most part, it does a pretty good job of getting all the jobs on the NJS queue, but we can bump it up if it requires more. And then as far as limits for a user, we don't have any limits for CPU usage or memory usage exactly. So you can theoretically have as much CPU time as you need. The only limitations are that we use a fair sharing system. So if the resources are being used by a lot of users at one time, then we try to allocate resources to those who haven't used as much, so that one person is not monopolizing the whole system. And so I don't think we have any more unanswered questions in the last couple minutes. Does anybody have any other questions? You can unmute if that's easier than typing. Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.